Good morning, everyone. Okay, so let's scooch up here. Okay, so what we're going to be studying today is something called, oh, hang on, let me just close my office door. There we go. On me. Something called transposons, right, which is kind of a collapse of the word transposable elements. Now, transposons are something pretty cool, right? Their kind of colloquial name is jumping genes, right? So they are mobile genetic elements that can move around independently within your genome, right? And are probably doing so to some extent right now, right? So this is I don't know, this is a kind of a really cool example of a, how can I put it? Like a challenge to existing dogma, right? So I'm always interested in really any kind of discoveries which turn existing scientific dogma on its head, right? And so we're very much, scientists, and even though we're often at the cutting edge of discovering stuff, we're also in many ways a very traditional uh, kind of group of people that, once we've got our things down pat, we kind of don't like that being changed, right? And so really up until the 60s, inheritance and genetics was really thought to be very much Mendelian, right? So, you have your chromosomes, you inherit one set from one parent, one set from the other, and yada yada, right? I don't think even at that point we knew that there was maternal only inheritance of mitochondrial DNA, for example. Anyway, so into this mix was I can't remember if she was a postdoc. I think she was a postdoc at the time, a lady called Barbara McClintock, right? And she was working on the genetics of maize, right? So maize is actually, you've seen it in the lab. It's actually a pretty cool organism to do genetics with because you get an awful lot of progeny, those kernels, on one cob, right? So you can gather a lot of data from one individual plant very quickly, right? Which is uh, a boon when you're trying to work out kind of complex genetics. And so what she found was that when she looked at a particular breed or strain of corn or maize or whatever you call it, um, basically she saw variable pigmentation, right? Even within kernels, right? It's like spots of darker color, and none of that matched any kind of Mendelian inheritance pattern, right? Actually, even within the same plant. And she really struggled to kind of explain why she was seeing what she was seeing. And so through a whole bunch of different approaches, she basically came to the conclusion that it was a piece of DNA that was moving around independently of the chromosomes and was interrupting. I'm not sure if it was a sugar processing gene or a pigment processing gene or something, randomly in individual cells, right? So it wasn't that you get a whole kernel that would be dark and a whole kernel that would be yellow. You get all kinds of intermediates all over the place. And this wasn't something that was very well received, to be honest. She struggled for several years to kind of gain acceptance. And now obviously it is dogma of its own, right? So it's now kind of accepted fact. And she got a Nobel Prize for that discovery a while ago, which is also really, really cool. And so transposons are really kind of a very alien concept. They're almost, one could think of them as being like genetic parasites, right? That kind of replicate and grow in our genome without really any input from us, like, kind of messing around with my DNA without my say so kind of thing. Let me bring up the chat. Oh, good question, Jelani. Yeah, it's very different. Mitochondrial DNA is essentially the same as bacterial DNA. So mitochondria have a haploid genome, and their DNA is circular. And also, there aren't actually all that many genes. I can't remember off the top of my head, something like 15 to 20 genes. So not all of the proteins used by mitochondria are produced by the mitochondria from its own genome. Most of them are imported from the cytoplasm. So during DNA isolation, it would be hard to get enough of it to be able to see it easily on a gel. But if you were to run it on a gel, and you were able to separate chromosomes and mitochondrial DNA by size, then the mitochondrial DNA would be a lower band on the gel. Unfortunately, for those kinds of sizes, it's actually quite hard to get good separation for really large bits of DNA in the kind of millions and hundreds of thousands of base pair size. So on the kind of gels that we'd be running DNA on for our lab, you wouldn't be able to distinguish between genomic and mitochondrial DNA. But you would be able to if you ran a much lower percentage gel, maybe. Never actually thought about that, to be honest. Anyway, cool question though. So yeah, essentially these are small genetic elements that can move around, right? And if you remember from our last lecture, we were talking about how a large percentage of our genome is made up of repeat sequences, particularly in the kind of middle repetitive sequence. Those are present in tens to hundreds of, maybe thousands of cases. Transposons are one of the main causes of that. Right, and because this happens over time, essentially, the older you are, evolutionarily speaking, the more of this stuff you accumulate. So, what are transposable elements? Right, essentially, they're mobile DNA elements. There are a huge variety of them. Right, really, like more than I know of, to be honest. Right, I really don't have a complete. I even worked on transposons for a couple of years when I was doing a postdoc in Australia, trying to genetically engineer a parasite to sterilize possums. Not opossums, those ugly little rat things that you have here, but kind of cute but vicious, fluffy possums that you have in Australia and New Zealand. But even then, I really don't fully grasp like the full, panoply of transposons that are out there. So basically, the best way of describing a transposon is essentially a piece of DNA. It has what are called terminal inverted repeats. These are important for transposition, right, for it to move. And functional ones, at least, have a gene in the middle which encodes an enzyme called transposase. This enzyme essentially recognizes a target sequence, cuts it, and inserts the transposon into it. Right? And these transposons make up not necessarily a majority, but a very large minority of our DNA. Right? And they insert all over the place. They actually have specific targets, which is unique to each transposon. But those targets can be present really high frequency. One that I worked on a transposon called piggyback, of all things. Its recognition sequence was TA. So anywhere that there's a TA in your genome, which there would be an awful lot of them, that transposon could insert itself, right? And this is the basic mechanism of transposition, right? This does vary from transposon to transposon, which we'll see in a little bit. But essentially what transposons do is their enzyme recognizes a specific sequence, TA for piggybacks, and it varies from being fairly short, 2 bases, 3 bases, to being up into the 8 or 9 base range, right? And so obviously the longer the target sequence, the rarer it is in the transposon. Kind of makes sense, right? Because these things occur essentially randomly. Now, what the transposase does is it creates a double-stranded break. But this double-stranded break is different from what you've seen before, right? It's not a blunt-ended double-stranded break where the 2 strands are broken like so, right? They generate what are called sticky ends, right? And so essentially what you have is 2 strands of DNA, and it cuts on one side, one strand over here, and the other one further down, right? So essentially you have 2 bits of DNA which would normally overlap and stick together, hence the name sticky ends. But now we're going to insert our transposable element, our transposon, into that break, right? But it doesn't have matching sticky ends to the host genome. It just goes in with blunt ends. And so this strand is then religated by transposase, as is this one here. And now what that creates is essentially this single strand of DNA on both sides. And so that's obviously bad. That triggers a DNA repair mechanism. And so DNA polymerase comes in, fills those gaps in, and then religates the broken strand on either side. And so what this does is it generates what are called flanking direct repeats, right? And essentially, if you look up here, right, we've got the sequence between the cuts is TCGAT, right? It's a 5 base sequence. Once the transposon has been put in and the damage it causes has been fixed, now you have 2 of those repeats on either side, one on either side. And so now you got TCGAT and you got TCGAT. This is one of the main reasons why transposons are so mutagenic, because not only do you insert a large chunk of DNA, and this can be, I don't know, they vary in size from about 1,500 to 2,000 base pairs to, I think, varies. I'm not an expert on them, like say 10,000 base pairs, 10 kilobases. Right. So obviously having that huge wedge of DNA land in a gene is going to disrupt it. But even when that transposon leaves, it leaves behind these flanking direct repeats. And so essentially what you have is a duplication. You have an extra 5 base pairs. So if you just kind of put your finger up and kind of put it over that orange bit on your screen, right? That's what the DNA would look like when that transposon leaves and jumps somewhere else. That's called an excision footprint, or it's the footprint of the transposon once it's left. So you can actually look at a genome and go, oh, the transposon has been here, 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 right? Because that's where you get these tandem repeats left behind. And so that, if that is in a coding region, that will be extraordinarily disruptive. That would almost certainly result in a loss of function for that protein, for that gene. And also now you can see not only do we have like large bits of repeat DNA throughout our genome, right? Because we have multiple copies of each transposon. We don't just have one that's kind of hoofing it around. We've got hundreds, if not thousands, of those. And I'm not entirely sure where they even come from, to be honest. I think they're kind of remnants from some cases retroviruses like HIV and other kinds of integrative DNA viruses. I'm not even really sure where transposons originally came from, right? Because it's not like they're out in the wild waiting to hijack an organism that wanders by. So anyway, these repeats, these tandem repeats, you can imagine, okay, well, next up we've got another transposon comes in here, and then we get now 3 of those repeats, and so on and so on. And so over time, you can build up like fairly large swathes of a chromosome that consist of nothing but these tandem repeats that are left behind by transposons. Another kind of characteristics of transposons is what these called terminal inverted repeats. And so essentially, what transposase does is it recognizes the ends, right? And these are called inverted repeats because if you read this one, ATC GCA, from left to right at the top, if you read it from the bottom right to left at the bottom, is going to be ATC GCA, right? That's why they're inverted repeats. And what it does is it basically kind of pinches up the transposon, kind of cuts it out, carries it as a loop, and then sticks it into another cut somewhere else. And so these inverted terminal repeats are really important for transposase to recognize that piece of DNA and kind of pinch it out and put it somewhere else. Now, another fact that can lead to essentially large amounts of kind of cruft or just junk in our genome is, we accumulate mutations randomly, both through cell division and through external insult, right? Radiation or, crap we eat or smoking or whatever. And those mutations occur pretty much randomly throughout our genome. They're not necessarily terrible, right? But if one of these inverted repeats gets changed, or if the open reading frame of the transposase accumulates mutation that knocks it out, essentially this transposon dies. It's kind of sad, but that is no longer a transposable element. It just kind of sits there, right? Kind of genetic baggage, I guess one could call it. And so if you think about this happening over a long period of evolutionary time, we're talking, tens to hundreds of millions of years, then you can see how, again, the more evolutionary old you are, I guess, then the more of this stuff you're going to have just lying around in your genome because there's not really any way of getting rid of it. It's not like you can do a spring cleaning. Well, that's a really good question, Jelly. Potentially, yes. Hang on, let me have some coffee. So I was actually, for a long time, I was thinking that transposons are essentially either neutral or negative, right? They either don't make any difference because they're landing in the middle of nowhere and they don't make a change, or they land in a coding region or a regulatory region and they screw things up. But I was actually reading the other day, I don't know how I got to reading about this, but basically an article about how, I think it was in Science actually. Get it on my iPad through the library, which is kind of neat. Basically, these regions can serve as DNA binding. These are binding sites for proteins, right? In particular, transposase, but they can also serve as binding sites for other proteins. And so there are several potential ways in which transposons can actually promote beneficial change or promote change which is advantageous, right? Because obviously this is random, right? And there has to be a benefit to having this change for it to be selected for, essentially, over time. But they can add new DNA binding, essentially regulatory regions to genes by bringing in these protein binding domains, right? And so other proteins can come in and then activate gene expression potentially, right? They also can create new genes by something called exon shifting, right? So where, eukaryotic genes are made up of a series of blocks called exons, which get kind of spliced together, which we'll talk about, when's that? Probably next week, to create a functional mRNA. Well, it's actually been shown that transposons have manipulated that by either adding a new exon or taking an exon out, so that changes the protein that's produced by that gene. And so if you have more than one copy of that gene, right, through gene duplication or something that's happened in the past, you can actually gain a new function for that gene, either through changing its expression or changing its actual structure of the protein. And so there's actually quite a lot of thought that transposons have actually driven a lot of evolutionary change over the years, right? And it's kind of really hard to get your head around, to be honest, but it made sense when I read it. Also, another thing that's kind of cool as well, not necessarily perhaps in humans, but certainly in plants, is that there are a lot of different organisms that live in plants, fungi, viruses, and even transposons, which confer stress resistance. And so you won't see the benefit of having that particular thing or DNA in your genome until you undergo some kind of adverse event or stress, in which case it becomes beneficial, right? So that's one of the reasons why I'm really leery of calling junk DNA junk, right? Because it's really just DNA that we don't know what its function is. It may actually have no function, right? We don't actually know because we don't know, right? But it also may have function that we haven't been able to figure out yet, which is really important in specific contexts, right? Kind of prolonged starvation or, prolonged heat stress or something like that. And so these transposons and other funky things like retroviruses and other things that kind of insert themselves into our DNA can actually potentially give us extra traits which we didn't otherwise have before. So it's really cool. I mean, I'm really not much of an evolutionary biologist by any stretch, but kind of thinking about how, traits change and are accumulated over time, I think is really fascinating. Particularly when you take the actual time element into account, which is pretty mind-blowing. Okay, so there are also a whole range of different types of transposons, right? So there are replicative and non-replicative transposons, right? So, for example, this would be if this transposon was then cut out and jumped somewhere else, that would be non-replicative transposition. Right? If this transposon was copied and its copy was stuck somewhere else, that would be replicative transposition. Right? So essentially, replicative transposition increases the number of those transposons in your genome over time, because it's essentially like you have well, it's like having a virus almost, right, in your cell, right? This bit of DNA is able to replicate itself and, go in different places. And so you'll accumulate that over evolutionary time. Non-replicative, you don't have an increase in the number of transposons, but you have an increase in the number of footprints left behind as it jumps around in your genome, and all of the various changes that that can induce as a result. So DNA transposons tend to be a little bit more common in prokaryotes and bacteria. And in eukaryotes, we have something called retrotransposons. And so retro basically means going from RNA to DNA. And that's why HIV is called a retrovirus, right? Because it has an RNA genome which is converted into DNA before that DNA gets inserted into the genome. Many people don't actually know that HIV is it integrates into the patient's genome, right? So you can be HIV negative for a number of years where you don't have active virus in your, I guess, in your bloodstream, but it's still present in your genome and it can get reactivated later. I think shingles is another one of those. Can't remember the virus that causes shingles, but that's again a similar kind of deal. My grandmother, my dad's side, used to have that. It's really quite unpleasant, by all accounts. Oh, there you go, type of herpes virus. That's cool. But I know that it kind of hides in a dormant state. I can't remember if herpes is a retrovirus or not. I'll have to look it up. Make a note. Anyway, essentially, what they do is they make a copy of themselves, but that copy is via an RNA intermediate. And then that RNA intermediate then gets made back into DNA, and that's what gets inserted into the genome. So essentially, this is what a retrotransposon would look like. Again, you have your flanking direct repeats either side that are the result of prior transposition, right? That's kind of the footprint that it created. And then as part of the actual retrotransposon, you have both transposase and also an enzyme called reverse transcriptase, right? Which is again also found in the in HIV. And so this whole transposon is transcribed, I believe, using the same machinery that transcribes other genes in the genome, right? The transcription machinery. And then once that RNA is produced, it gets reverse transcribed and basically turned back from RNA into double-stranded DNA. Typically, actually, what happens is you get a single strand of RNA gets turned into a single strand of DNA called complementary DNA or cDNA, and that then gets copied to make our double-stranded DNA down here. Now, this is then a free genetic element that can be inserted wherever. And so now we get the same deal happen again. We have the staggered cuts made by the transposase. The double-stranded DNA gets inserted into that double-stranded break. Pardon me. And as before, we get a DNA repair mechanism kicks in and it fills in those gaps and creates new flanking direct repeats on either side. So now you can see that now we have like multiple copies of this transposon in our genome. And some of these copies will die through mutation over time, but they'll still be able to kind of move around. Actually, in terms of kind of using transposons as a tool, you can actually take think flies, yeast, and even early in C. elegans biology, scientists actually use naturally occurring transposons to do like forward genetic screens, so to mutate the genome of their, target organism, and then look for mutations, right, to be able to kind of figure out the function of different genes and different processes. So we could actually use that. My work was to, well, the idea behind it, it didn't get as far as we wanted, unfortunately, is for some transposons, you can actually cut out that inner part, take away the transposase, and put something of your own in there, right? In my case, it would have been a small protein that sterilizes possums, basically. And then you can use that with transposase that you add yourself, right, at the same time to stick that into your target organism genome. Unfortunately, it didn't get very far. I got shit canned by the New Zealand Research Authority, unfortunately. So transposition is actually always mutagenic, right? I'm not sure why often is there. Here we go, let's change it. Always. Right, because it will always change the DNA of the site that it gets inserted into, either through its presence, because it's large, or because when it moves again, it leaves behind that repeat that didn't exist there before, right? Which will essentially is an insertion mutation, right? It's a creation of new bases that didn't exist there before, right? And so, as I said earlier, you can really use this as a tool, right, to generate mutations in your target organism. And this was what people did in Drosophila an awful lot. And actually, they're able to use that as a means of creating transgenic Drosophila, right, via the mechanism that I just talked about. And if that occurs in or near a protein coding region, that can, as I said earlier, result in a knockout of that gene, depending on exactly where it inserts. Or in probably rarer cases, it can actually create new genes or new regulatory elements that control those genes. And obviously, a lot of the time, it will also just do nothing very much, right? It'll disrupt wherever it's going, but where it's going doesn't necessarily have to be useful. And also, cells actually have means or ways of suppressing the activity of transposase because it is so mutagenic, right? That, you know, that's not necessarily a good thing, right? So it gives you a selective advantage essentially to suppress transposition. Also, another neat thing which kind of leads to really large scale changes is that basically you now have multiple copies of the same bit of DNA throughout your genome, right? And those multiple copies can now serve as sites of homologous recombination. Because again, with homologous recombination, the aim is always to, you know, kind of line up bits of DNA and cross them over where they match exactly, right? So typically that only occurs between non-sister chromatids of homologous chromosomes. But now if you have, you know, let's kind of put it around the other way, a matching bit of DNA over a big enough stretch, right, between one chromosome and another, now you can get those lining up, right? And now what happens is that you get a crossover between different chromosomes, and you can actually get a fusion of different chromosomes. That's happened at least once in the evolution of chimpanzees, for example. Can't remember which chromosomes, but if you compare chimpanzee genome to human genome, basically two of our smaller chromosomes were fused together in the chimpanzee lineage, right, from our last common ancestor. And also another funky thing that can happen too is that if you have within the same chromosome multiple sites which are identical, When you get recombination, now you can actually flip part of the chromosome, so it's in a completely different orientation, right, called an inversion. And when you look at closely related species, right, so you can do this for, Cenorhabditis species for worms, or, compare, humans, gray apes, and chimpanzees, for example, you'll actually see, even though they may share the same chromosome, parts of that chromosome will be in a different orientation. Same genes, just not in the same order anymore because part of the chromosome has been flipped over. Okay, anybody got any questions about transposons? We're pretty much done there. So inversion does really funky things, Rogelia. So for the genes which are kind of in the middle, doesn't make any difference by and large, right? Because they're still in the same kind of context. If you think about swapping like one whole arm of a chromosome round, it might not make any difference. But for those near the edges, essentially, and this is where we get into control of gene expression, right? Essentially, you have a gene here, and that can be controlled by a piece of DNA. Let me show my hands in view, a long distance away, right? Doesn't have to be very close to that gene. It could be, potentially hundreds of thousands of base pairs away. Now, if you kind of flip that round, right? So that's now over here. It is no longer in the same context. Now you actually might have a different gene come up here that now is going to be in the position to be controlled by that genetic element. And so it can be extraordinarily disruptive. Essentially, you're changing the context of a whole bunch of genes, which can change their regulation. If a gene happens to be right in the middle, obviously it will get cut in half. You can have things called gene fusions, which are often also facilitated by transposons, where you get part of one gene, say the promoter, like the regulator region, and a little bit of the protein, and the butt end of another gene, right? So you have what's called a chimeric gene, right, between two different pieces. And that is often a very bad thing to happen. That's has been thought to be the basis of several different types of cancer, because essentially you gain like a new function which you might not really want. So it very much depends. For a bunch of the genes, no, it's not going to make any difference. For some of them, it can make a really huge difference. It just depends, right, on what's moving and what it's leaving behind or what it's gaining. Yeah, but again, depending on what happens, some of those changes may actually confer a selective advantage to that individual, such that it's more likely to survive and reproduce and pass on its genes. And so then, that gets selected for in the population, and then it becomes fixed, right? All individuals in that population end up having that change. So that's definitely the kind of thing that you'll learn more about when you take evolution. Probably with Liz Border, I think she's the one that teaches that. But it's kind of funky to think about like these really large scale changes occurring, right? Whole scale rearrangements essentially. Okay, so kind of coming back up here. Pardon me. So I'm guessing your question is, what causes the transposon to stick to this bit of DNA here and this bit of DNA here? Is that right? Well, that's because the transposase has a form of DNA ligase activity, right? So it's able to join or basically create a phosphodiester bond between this base on the kind of host strand and the adjacent base on the transposon. And it's able to do the same over here. right? So transposase can both cut DNA, right? So it's a endonuclease, but it's also a ligase as well because it can stick that bit of DNA into it. And so this top strand here is now one continuous piece of DNA all the way to the end of the transposon over here, where there's now a gap. And the same for the bottom strand. It's continuous all the way from the host DNA into the transposon to the end. Then, so that's now stuck, right? So now it's not a double-stranded break. It's the chromosome is kind of stuck back together. It just happens to be a transposon in the middle of it. But then, obviously, this single-stranded DNA has to be fixed because you can't leave that there. And so that's where the DNA repair mechanism comes in, fills in the gap with DNA polymerase, and then another DNA ligase will come in and seal the backbone by creating a phosphodiester bond between that new flanking repeat and the transposon. Good question, though. Does that help? Yeah, essentially. So it's basically break it apart, stick it back together, and then rely on the host cell to fix the mess that you've created, so that you'll end up with a intact piece of chromosome, intact piece of DNA, but now you have an extra sequence, one of these flanking direct repeats that you didn't have before. All right, so I don't have any more questions. Let's just double see. Okay. You know, we have actually plenty of time to talk about gene expression on Friday. So I'm going to just call it a day for today so I can kind of have another cup of coffee and catch up on some stuff. And we'll start on gene expression on Friday. So stopping a little early, but that's not necessarily a big deal. We've got plenty of time in the schedule. And some of you I'll see later in lab, and some of you I will see on Friday. So have a lovely day. Enjoy your extra 8 minutes of freedom, and I shall see you soon.